Good morning, everybody. My name is David Thwaites. I'm from Newcastle University, as Paul said. I'm here as a representative of the Physiology Council, from which I'm going to stand down in about 2 hours' time. So I can do whatever I want, and they can't stop me. So I can say whatever I want. This is the last job I've got to do, and it's one of the good ones, and that's to introduce this year's Joan Mott Prize lecture. I'm delighted to say that the recipient this year is Hannelore Daniel, who's sitting over here. Hannelore is the Professor of Physiology of Nutrition at the Technical University of Munich in Germany. She's had a long, and I think probably longer than both of us would like to think about, but very distinguished career in the area of physiology, biochemistry, molecular biology and omics of nutrition, and I think that she's one of the people who've probably created the area which is now called molecular nutrition. Did much of her training in the University of Gießen in Germany. She rose from a diploma student all the way up to being the professor of the biochemistry of nutrition. And during that period, she also spent time in the physiology department in Glasgow, working on acid microclimate, and also time in Pittsburgh, working with CM-ACADB with brush border membrane vesicles. In '98, she moved to her current institution to take up the position of Scientific Director of the Institute of Food and Nutrition Research, and she's been there ever since. She's often in big demand, and I know she's been made lots of good offers to go into industry from there, but for better or worse, she's stayed where she is, and she's going to tell us about the work that she's done there over the last few years. She's won many different awards and prizes. I'm not going to bore you with listing those today. I think her preeminence is evidenced by the fact that she has, since 2011, been the chair of the EU Commission on a Healthy Diet for a Healthier Life, which is probably something that we should all take perhaps more interest in that we do. I first met her at the IUPS meeting in Glasgow in '93 at the poster session. And I was a young postdoc. She was already very well known by that time. I went and introduced myself, and after a few seconds, we were surrounded by 4 very large men in dark suits, shaven hair, reflective glasses, earpieces. It was a bit like being in Men in Black. Initially, I thought that perhaps this is how she always travelled, with her own sort of security guards. But then I could see in her face that she was equally disturbed by this as I was, because these people weren't saying anything to us at all. But from behind her left ear, I noticed the telltale shape of the House of Windsor nose. And it turns out that Princess Anne had stopped by to look at the poster next to us. I didn't realise she was so interested in organic ion transport, but there you go. You learn something every day. So since that unusual beginning, we've remained colleagues and collaborators and occasionally competitors. But as you'll see from her talk, it's been a very one-sided competition. So without further ado, I'll introduce the talk, which is Nutrients in the Gut from Moving and Sensing. So Hannelore.
So, good morning, everybody. David, thank you very much for this lovely introduction. I can call myself a lucky person to have colleagues like you talking so nicely about me, because here and there I can be pretty nasty. Now, of course, I have to thank the person who put forward my name, whoever this person was. I guess I have to thank a committee. And of course, I have to thank the society for bringing me here and to be on the stage now. Thank you very much. I didn't know too much about Joan Mott, and actually I couldn't find too much. But I stumbled over this quote here, which tells me if I would have got to know her personally, I probably would have liked her very much. A person that stands up for something, that goes for it. She probably was a nasty person as well, being a little bit stubborn. But I feel proud to receive this award. And if somebody would say, Hannelore Daniel was very direct and forthright, with wide interests and strong opinions, and she was generous to younger students, That would be good. So, younger people. When you're young, you may have a jigsaw puzzle. And I put that up because it reflects my entire scientific career. 35 years in experimental research, looking into intestinal functions and transport of nutrients. It is the most rewarding thing when after 10 years, 2 pieces of a puzzle come together and give you a flavor of the picture. So, continue in science, and you can play. You have your puzzling area, but be aware, there's a major difference, well, there are actually 3 major differences between a jigsaw puzzle that you played when you were a child and a scientific game. The first is you don't know how many pieces are in the game. The second is there is no box that shows you the picture. And the third one is you most likely never finish it in your lifetime. But in between, you can have fun. So I hope I can share a little bit of the fun that I had. I will focus on 2 aspects. And I guess my presentation was put into place just before lunch to teach you what's going to happen in your GI tract. I hope I'm not screwing you up in terms of losing your appetite. I still enjoy eating, as you can see. And I will pick 2 pieces of the puzzle. And one is my most beloved protein on this planet, because I've been characterizing peptide transporters for 25 years. And then a new aspect of nutrient sensing. And that brings me actually back at the end of my career to my beginning, because I started in 1978 in looking into glucose transport processes, and now at the end of my career, I'm looking into glucose transport processes. So, amino acid and peptide absorption, 2 aspects give you a little bit of an overview, and then I show you that moving peptides is hard work. Now, lunchtime, okay? You eat protein. The proteins are chopped down. There are various enzymes secreted by the pancreas. And then you also have various enzymes attached to the apical membrane of the brush border. Here, the amino-oligopeptidase, aminopeptidase is releasing free amino acids, but also providing short-chain peptides for uptake into the epithelium. So the epithelium is equipped with a whole array of amino acid transporters, ion-coupled, but also exchangers. And there is my most beloved protein, peptide. Now, almost each of these proteins mediating amino acid import and export, whether on the brush border or the basolateral membrane, has malfunctions in inherited diseases, such as the heart-nap, aminoglycinuria, cystinuria, etc., etc. And the first evidence that in addition to those amino acid transport systems is a system that is capable of transporting short-chain peptides was derived in the late 60s and 70s based on observations in patients suffering from those diseases. And I give you just one example. And this is here Eddie Hartnett, the patient that showed this huge elevation of amino acid levels in urine because of a transport defect in the renal proximal tubule. Now, the same protein that mediates transport in the renal tubule is found in the intestine. So this malfunction causes loss of amino acids, and the corresponding malfunction in the intestine should cause a loss of amino acid absorption. Well, that is a severe disease. I don't want to go into details, but here is interesting evidence in those patients, both Hartner patients as well as patients with cystinuria. This evidence is derived from perfusion studies in the human intestine. Now look into literature here, 1970s and 1971. I strongly recommend for the youngsters to look in a little bit of antique literature. There is damn good science to be found. So here's the evidence. We look at the serum histidine levels in a patient, the Hartner patient here, in a healthy controls. Over time, after ingestion of either a mixture of beta-alanine and histidine, those two amino acids make up the carnosine, which is then the dipeptide. And if you have the carnosine provided, you see that the serum histidine levels increases. But if you have the amino acids in the patients provided in a one-to-one stoichiometry, you see there is in essence no increase in histidine level. So there is clearly a malabsorption of histidine. But there is clearly also an increase in histidine levels when you provide it as a dipeptide. So obviously dipeptide uptake into intestinal epithelial cells can overcome here this malabsorption of histidine. In the healthy controls, uptake is a little bit higher, but that obviously is sufficient to provide even essential amino acids, because this transporter defect would also cause a malabsorption of essential amino acids. Now, how can you live and how can you grow if you have a malabsorption of essential amino acids? Well, the good message is, as long as you have a peptide transporter, you're on the safe side. And the same holds true here for this cystinuria patient, where glycine and lysine was provided either as a mixture of amino acids or as a dipeptide, as glycylysine. And lysine, again, is essential. So if you perfuse the intestine with the amino acid mixture, you clearly see that the patient here has a malabsorption. But if you provide the whole amino acid mixture here as a dipeptide, you see you have almost normal absorption. So that was the best evidence that there is a transport system capable of taking up short-chain peptides with release of the amino acids in the cell and then translocation into systemic circulation. So that is my protein of interest, as I told you. Isn't it a beauty? So that's a little bit of fantasy, based on some crystal structures of bacterial proteins, which have very similar activities, despite the fact that they only have about 20% homology. Now, membrane, cytosol contains amino and carboxy terminus, and then we have a huge extracellular loop of more than 200 amino acids projecting into the intestinal lumen. And we have no idea what this loop is good for, because it is only found in higher organisms, not in bacteria. And I'll come to this loop in a minute. Now, peptide-1, the intestinal peptide transporter, is a fascinating protein. It transports almost all possible dipeptides and almost all possible tripeptides. Take 20 proteinogenic amino acid, makes 400 different dipeptides, 8,000 different tripeptides, and you even can modify those. There are literally thousands and thousands and thousands. That is weird if you think about specificity. Usually we think biology has to be very specific. So is this a garbage can? The answer is no. It is very specific. You could call it specifically unspecific or unspecifically specific, because it is selecting substrates in a very stereospecific manner. It even can discriminate whether you have a cis or a trans peptide bond. It doesn't take anything that is longer than 3 amino acids. It doesn't take any free amino acids. It always is electrogenic, which is even more bizarre than substrate specificity, because you can have anionic or cationic or switterionic. It always generates an inward current. It transports small and big ones, hydrophobic and hydrophilic ones. And it is damn difficult to understand how a single protein cannot do that. Now, it is also an acid loader, and that kept David busy for quite some time. How can epithelial cells then cope with the acid load? Well, they have sodium proton exchangers, and in particular NH3. And there are more people in the audience working on this protein. And that is the pharmacological or pharmaceutical dimension, because the peptide transporter, I have to admit, is a little bit stupid. It doesn't know whether it is a real nutritional peptide or a peptidomimetic. It looks at the structure, of course. And you can smuggle in drugs. So you have aminocephalosporin antibiotics that are transported. You have some selected ACE inhibitors and more prodrugs designed to match the substrate binding domain, because then you have almost 100% bioavailability. So there's a lot of interest in pharma and peptide transport. Now, this is my SU, which means we look into peptide transport processes in everything that lives on this planet, starting with bacteria. So we're cloning, we express, we purified, and try to crystallize bacterial peptide transporters. So there are 4 related proteins in E. coli. I got into C. elegans about 10 years ago. We knocked out the intestinal peptide transporter and phenotyped the animals, and this was a very interesting exercise, because we learned that the peptide transport in the intestine has something to do with the capability to cope with ROS, reactive oxygen species. When we cross it into an insulin receptor deficient mutant, we ended up with a Methuselah worm that lived 4 times as long as wild type. Completely unexpected. Just showing you that amino acid bioavailability is linked to aging processes. We cloned some fish transporters, from salmon to zebrafish, but the most interesting one was the ice fish, captured in Antarctica, an animal that lives at temperatures where normally biology doesn't function. And the transporter from the ice fish had a temperature optimum at 2 degrees centigrade. And it even worked at -2 when everything is frozen. And you ask, how is this possible? How can a protein operate at very low temperatures? This protein was in essence dead if you heated it up to 37 degrees. And funny enough, we found a stretch of amino acids, 7 amino acid residues in the carboxy terminus that you only find in this Antarctic fish. And it confers the cold adaptation, because you can surgically, or protein chemistry if you like, transfer this stretch of 7 amino acids, and you transfer the temperature dependence. You can turn a warm-blooded peptide transporter cold and the other way around. That is really funny. So here is the most painful exercise in my entire peptide transport research career, because based on the C. elegans, we were expecting a nice phenotype by knocking out the peptide transporter in mice. And you put a mouse on a standard high-carb diet, and the mouse is happy. Hannelore Daniel was very unhappy. And then we started feeding low-protein diets and high-protein diets and low-fat and high-fat diets. And I can tell you, one knockout produces 4 different phenotypes, depending on the diet. And I learned that when you feed a black 6 mouse that doesn't have a peptide transporter, a high-fat diet, it stays slim. I immediately got a patent on peptide transport inhibitors. I tried them myself. It didn't work. So, and finally, some studies in humans relating to the pro-drug approaches. We were involved in looking for the bioavailability of some DP4 inhibitors, and some of them even made the market, and they have excellent oral bioavailability because they utilize the peptide transporter. So, a delicacy. Moving peptides is hard work. Now, this all started with a strange observation. That is the large intestine of a mouse. Immunostaining for the peptide transporter in the small intestine, which would be up here. And you see wonderful staining of the brush border. And then we dissected the colon and learned then that in the far distal colon, even in rectum, peptide transporter comes up again. But there is nothing in the proximal colon. And we have no idea what it is doing in the distal colon. But what's even more striking was this observation here. When you did a Western blot and you compared the mass of peptide-1 small intestine and distal colon in a black 6 mouse or even in a germ-free mouse, you always ended up with a higher molecular mass for the protein in colon than in the small intestine. Strange. The same protein, small intestine with a lower mass than in distal colon. And if you chop off the glycans, that's done here, yeah, you bring the protein down to a mass of about 60 kilodalton, which means that you have about 35 kilodalton of sugars attached to the peptide transporter. That's quite a lot. So, glycans. What do we know about the glycans in glycoproteins, membrane proteins? Actually, not too much. Now, peptide has, in the extracellular domain, 6 putative and glycosylation sites. And we got interested which glycosylation site is used for glycosylation and what is the role of the glycans in the peptide transporter. So this is work by my PhD student Tamara Stelzel, and she did a wonderful job here. Because what she did is she deleted each asparagine in those consensus domains for glycosylation, individually and collectively all. And what you see is a stepwise decline in molecular mass down to about 60 kilodalton, the naked protein. All those proteins lacking individual asparagines and glycans, or lacking all the glycans, made it to the membrane. So we could even characterize their function. And we did electrophysiology, 2-electrode voltage clamp. I told you the transporter is electrogenic, it moves protons, and it generates wonderful currents. When we use these different mutants, either the individual mutants or the multiple mutants, something really weird popped up. Now look here. There is a mutant at position 50, exchange of an asparagine to a glutamine, and this showed a 2- to 3-fold increase in maximal transport currents. Huh? How is that possible? How do you explain a gain of function like that? Well, even more bizarre was that was associated with a loss in affinity. Going back, or you can see it here, whenever, even in these multiple mutants, whenever we have a N50Q, you see this increase in transport currents. Now you have to believe me, we did surface biotinylation. The density of the transporter, this N50Q mutant, is identical to wild type. So it's not more protein in the membrane that causes this increased transport activity. That is really weird. So what we did is we also used tunicamycin, preventing glycosylation in the oocyte. And again, the transporter that was then going to the membrane without glycans was faster. And that was not only in the mouse, it also was in the human. So whenever we mutated N50, we had increased transport activity, but a reduced affinity. Why that? Well, Tamara took advantage of a substitution of the asparagine by a cysteine. So we deleted the asparagine, no glycans added here, put in a cysteine, and then modified the cysteine with an MTSA biotin to simulate sugar structures in terms of weight put on N50. With the biotin, we have about 500 Dalton added here. Now, funny enough, as you see here, when we have the cysteine sitting there at 50, we have also the increase in transport activity. And if we put the biotin on, it goes down. So here is the current, and here is the affinity. So very similar to what I showed you before with the N50Q. So I think that is a good example that the velocity and the velocity increase when we chop off or prevent glycosylation at that site enables the protein to move faster. So we are currently looking with different weights attached to the position 50, how the weight affects the movement. So moving peptides is hard work, in particular if you have a lot of glycans sitting there. Now, let's change gears. You're not only eating protein, you also eat carbohydrates, you eat starch, you eat sucrose, and maybe some monosaccharides such as glucose and fructose. I will address the role of SGLT1 and GLUT2 in overall glucose absorption, mainly in knockout mouse models. And I also will address a little bit the mystery of fructose malabsorption. Now, again, dietary carbohydrates, tons of starches whenever you have your bread and your pasta, your rice or whatever. Alpha amylase takes it down, and then you have glucoamylase and maltase, and for the disaccharides you have the sucrase and lactase, etc., chopping everything down to the level of monosaccharides. Now, this is a famous guy, Robert Crane, for some physiologists probably known. 1960, he drew that in Prague as the first idea of how glucose transport is coupled to sodium movement. And of course, now we know that this protein here, SGLT1, is doing the job. So SGLT1 transports glucose and galactose. And next to it is GLUT5, one of the members of the GLUT family. That is the fructose transporter. That is textbook knowledge, okay? However, there is a problem. If you look at the affinity of SGLT1 in whatever kind of expression system, you end up with a very high affinity. And if you look at the old kinetic data from human perfusion studies of the small intestine, you see that the affinity is at least 10 or maybe even 100 times lower. So high affinity kinetics for the cloned protein and low affinity kinetics in the human condition in vivo. So what's going on here? Well, there was a hypothesis put forward, and there are at least two protagonists pushing the concept that the transport of glucose via SGLT1 causes an opening of voltage-gated channels here with an activation of signaling events in the cell, causing a recruitment of vesicles into the apical membrane, now with the insertion of GLUT2. And GLUT2 is a uniporter, which then would open the door for more glucose, but also fructose coming in. Now, that is the structure of SGLT1. Again, there is no crystal structure of the mammalian protein. That is Ernie Wright's work, with a bacterial homologue, with, let's say, best estimate of how the protein may look like. 14 membrane-spanning domains, and a good matching of experimental data with the crystal structure in terms of the pore-forming structure that allows the sodium and the glucose to move through. I got interested in fructose absorption because my PhD student, Kerstin, is afraid of eating 1 gram of fructose. And she claims she has discomfort, she has cramps, everything. And then I see her putting a whole spoon of sucrose into tea. I said, "What are you doing?" He said, "Why? I don't have a problem." I said, "You're telling me you have a fructose malabsorption with all kinds of problems." "Yes, I do, but I don't have a sucrose problem." Hey, come on. Yeah, sucrase, yeah, hydrolyzes your sucrose, and then you have fructose, and then you should have trouble. And again, old literature. So here we talk about new ones in 2011, but already in 1981, it was reported that you can get rid of the fructose malabsorption just by adding glucose. So here we talk about a phenomenon that is a little bit of a fashion disease, I guess. It become very popular. And it may be that we all are more exposed to fructose because we have many more fruit juices, and so the average intake of fructose is considerably higher than it was 50 years ago. And obviously the human gut is not made for handling large quantities of fructose. So there is a limited absorption capacity. And you can easily detect the malabsorption just by measuring hydrogen exhalation in breath after a load of fructose. And that was done in patients here, and that is a paper from 1988, giving 50 grams or 37 or 25 grams to people that reported fructose malabsorption and measuring hydrogen appearing in breath. And you see, as you increase the fructose load, you have more and more people that have a significantly elevated hydrogen exhalation because bacteria break down the fructose that was not absorbed in the gut. And here comes the observation. When you add glucose in the same volunteers or patients, the fructose malabsorption is gone. That is amazing. And that, again, is dose dependent, as you can see here. So, the secret in preventing, or at least having all the discomfort associated with fructose malabsorption overcome, is take a spoon of dextrose. Now, this is around for 30 years. The mechanism is unexplained. Now, coming back to the model here that I just presented to you, yeah, the inclusion of GLUT2, of course, would be an intriguing concept to explain what you just saw, because SGLT1 would bring in the glucose, and then you would have the GLUT2 coming into the apical membrane, and GLUT2 would open a second entry route for fructose. Now, we're lucky people because we have access to these 3 different mouse models, like 6 mice, either lacking SGLT1 or lacking the GLUT5 transporter or lacking the GLUT2. So how did we approach the question of which transporter is so important? Is there a trafficking of GLUT2? And if so, yeah, is GLUT2 insertion then allowing an increased fructose uptake? We used a gavage. So we have a certain amount of glucose that, when in the intestine, increases the intestinal glucose concentration above 150 millimolar. It was intentionally pushed to a very high concentration because previous papers had suggested that this GLUT2 trafficking only occurs if you have very high glucose level in the lumen. We not only gave glucose, we also gave radioactive glucose along to be able to measure the appearance of the radiotracer, both in the tissue as well as in blood. So the animals were sacrificed 15 minutes after gavage, and we removed the entire intestine and chopped it down centimeter by centimeter by centimeter, and measured first the amount of radiolabeled glucose in these individual segments. So you see the entire small intestine with 32 segments here. And you see when you have a mouse that lacks SGLT1, you have a markedly reduced accumulation of glucose. Yeah, so if you take the area under the curve here, yeah, there are about 15% of glucose uptake in this respect, radiolabeled glucose in tissue, yeah, when the SGLT1 is gone, confirming that SGLT1 is important. Not to say it is the most important transporter. Okay, if you look tracer in plasma, that's what you would expect, markedly reduced. And even the labeled and non-labeled glucose, you have a reduced increase in plasma glucose from the glucose load. So, confirming SGLT1 is important. Now, this is the GLUT2 knockout mouse. So, no GLUT2 means whatever kind of signaling you have, there can be no GLUT2 going into the apical membrane. But maybe you saw that GLUT2 is in the basolateral membrane, and in textbooks, yeah, it tells you GLUT2 allows glucose to leave the cell across the basolateral membrane. There was actually a paper published in PNS a couple of years ago by Stümpel showing that the animal that lacked GLUT2 didn't have any problem in getting the glucose out of the intestinal epithelial cell. Challenging textbooks. Now, we challenge textbooks. This is GLUT2. Now, it's a different picture, as you see. The knockout animals now have more glucose from the tracer in the tissue. How comes? Well, GLUT2 in the basolateral membrane is lacking, so you don't have an efflux. We confirm textbooks, at least a little bit, which means you have more glucose in the tissue and less coming into blood. And that's exactly what you see here, reduced tracer concentration and reduced increase in plasma glucose. So, this experiment didn't give the answer. But there was no GLUT2 in the apical membrane. Yeah, it doesn't prove that GLUT2 trafficking takes place, but it doesn't disprove it either. So we also did the detection of the proteins by Western plotting. The problem is GLUT2, to detect GLUT2 in the apical membrane in mouse is a pain. And we always end up with a little bit of contamination by basolateral membranes, as you can see with sodium, potassium, ADPase here. So in the end, the answer is, I really don't know. But I don't believe that GLUT2 plays a crucial role as a second apical transporter. Now, this is immunostaining. The antibody works quite well for SGLT1, and you see in the knockout mouse there is no labeling. And then the GLUT2, yeah, that is lateral here and that is basal. Their antibody is okay, but we never found evidence for, let's say, a certain level of GLUT2 in the apical membrane. Fructose. What we did here is, again, a radio-labeled, non-labeled mixture of fructose by gavage to wild-type animals or to GLUT5 fructose transporter deficient animals. And we combined the fructose either with alpha-methylglucoside, which is a substrate of SGLT1 and not of any of the GLUTs, or with glucose, which would be a substrate of SGLT1, but also of a GLUT2. Now, based on the concept in humans that glucose can overcome a fructose malabsorption, we were asking whether this co-administration of these two monosaccharides with the fructose increases fructose uptake. And surprise, yes, the glucose indeed increased the uptake of fructose into the intestinal tissue. Look here, this is the GLUT5 deficient animal. So the maximal level we reach here is about 100 nanomoles as compared to 5 to 600 here. So GLUT5 is the key fructose transporter. And yes, there is an effect of glucose, but not of alpha-MDG by increasing fructose uptake into the intestinal tissues. Now, changing gears, the title was moving and sensing, so we come to the sensing part. We have been moving peptides, we have been moving monosaccharides, and now comes sensing. And I put up 2 seminal papers here, a collaboration of Hermann Köpsel with the Frank Reimann in the audience and Fiona Gribble. Super research on the role of SGLT1 in sensing. And we also collaborated with the Cambridge people on sensing of peptides in the intestine. So the idea is that these transporters are not only in the epithelial cells and with the responsibility of moving substrates across the brush border, they are also found in endocrine cells, in L cells and K cells, with different spatial distribution along the gastrointestinal tract. And the new role is these proteins can also do sensing. And the sensing here means coupled to the secretion of the incretins, in particular of GIP and GLP-1. So, and if you do that in SGLT1 deficient animals or in wild type animals, you see after gavage that plasma GIP levels in the knockout animals are gone. Nice robust increase when you give glucose here, and in the knockout animals, no response. GLP-1 output also drastically reduced. Insulin levels in plasma increased by the glucose gavage and significantly reduced in response in the animals that lack SGLT1. And here we have the GLUT2 animals, either wild type or knockout animals. Glucose, nice increase in GIP, nice increase in GLP-1. In GLUT2 deficient animals, a little bit of a reduction here, no effect here, yeah, and a significant effect on insulin. The animals per se have a problem, and some of you actually should have shouted, "What are you telling us? GLUT2 deficient mice? GLUT2?" Well, there's a problem with the species, GLUT1 and GLUT2 in beta cells. Yeah, so GLUT2 is considered to be the key transporter in beta cells. So these animals had a GLUT1 re-expression in the beta cells. Otherwise, they would have even more severe problems. So the message here is, yes, SGLT1 is crucial for secretion of GIP and GLP-1 in response to an oral glucose load, but GLUT2 has, in essence, no function there. And the last small session is about growing little guts. So we've been starting using organoids to assess transport function and sensing. That is a wonderful tool, but very expensive. So you have to plan your experiments very carefully. Well, you anyway should very carefully plan your experiments. So you take your mouse or animal intestine with the stem cells and mince it and put it in a suspension, shaking, and do some centrifugation. And then you do an embedding here, and you have to add these specific growth factors, and then you end up with these interesting structures here. And the interesting structures resemble the small intestine, except that they don't have real villus structures, but they have beautiful crypts, and they also have mature enterocytes. So if you do a staining for villi here or eat caterin, you see, that is what you would expect. Everything is at the place where it should be. So the problem is they're closed, and the lumen is inside, like in a real gut. But the problem is, how do you get something into it? because they don't have an esophagus. So we were looking carefully in how we can get nutrients into the lumen here to then study the uptake from the luminal site. And the same thing holds true for sensing, because you also need to get your nutrients into the lumen before you can have a secretion of hormones out of endocrine cells. But as you will see in the next slide, combining now both things in an organoid, you can measure that. So immunostaining for SGLT1 shows nicely the brush border. There is peptide-1 in the organoid of the mouse, and there is GLUT2 with the basolateral location. In the organoids from the corresponding KO mice, you don't see any stains. So clear-cut orientation of the proteins as you expect to see. And that is uptake now of glucose in wild-type animals, and this is the most potent inhibitor of the SGLT1, the shell cone fluorescein, and you see a marked reduction. This is glucose uptake in organoids from SGLT1 deficient animals. You see, in essence, no difference to fluorescein inhibition, but if you add fluorescein, you still have a little bit of transport that you can push down. This is alpha MDG, the more specific transport substrate for SGLT1, because glucose could also be transported by another transporter. GLUT2 or GLUT1 or any of the other GLUTs. So here is SGLT1 specific substrate in the knockout, almost gone, and you see fluorescein doesn't do too much. So GLUT5, fructose uptake, in wild type and in GLUT5 deficient animals, and fructose with fluoretin, not fluorescein, as an inhibitor of GLUT5, and everything is like in textbooks. Down here, we talk about peptide transport activity. Now, if you look here, this is a pronounced effect, and here in the knockout animals that lack peptide-1, we still have a considerable uptake. And this uptake can be inhibited further by adding cefadroxil or any other dipeptides to the medium. Now, David looks at that is a good evidence that we talk about a basolateral peptide transporter, now accessible from the basolateral site. We know that there is a peptide transporter in the basolateral membrane, but it is not characterized on a molecular or genetic level. So that would be the next thing to do, phenotype what that part of the transport process here is in organoids. Okay, peptide-induced secretion. So when we measure GLP-1 in these organoids, and at glycosylsarcosine, a model substrate for peptide-1, we see a stimulation of GLP-1 output that is gone in the peptide-1 deficient animals, confirming previous collaboration papers. And that is really funny. If you do the immunostaining here, you have to have only one endocrine cell, and you can measure hormone output in these organoids. That is amazing. And we even do some imaging there, both for glucose responses in terms of calcium traces here, as well as pH changes with the classical fluorescent markers. So, that is the most important part. I'm just the one that talks about all that. Well, I have to bring in the money. These are the people that burn the money. But they are nice people. So this is my team. Not all of them work on transport processes. And I have put up the names of the people that have contributed to this stuff I showed you today. We have a lot of collaborations. And it has been a privilege to work both with those people here as well as all the partners from quite different sites and continents. I also have to acknowledge funding agencies, and over 25 years, the German Research Foundation has supported my work. And I wish the youngsters here that they can have an area of research where they have constant support over 25 years. don't build on the Commission. That is a political statement, yeah? Currently, funding in Horizon 2020 is really, really bad. So, anyway, I was a lucky person. And I'm a lucky person being here today, receiving this prestigious award. And I hope I could share a little bit of enjoyment and excitement for science with you. So, I was asked to put up that as the final slide. Thank you very much, and enjoy your lunch.
So, I'm Anne King, and it's my great pleasure and indeed honour to present the vote of thanks for this year's wonderful Joan Mott Lecture 2014, and in particularly because, as you'll be aware, we are celebrating 100 years of women in contributions to physiology and within the society. I listened in awe to Professor Daniel's lecture, and I was particularly struck, as I'm sure you were, by the sheer breadth of the techniques that were deployed and used in very smart ways with a diversity of different types of models, ranging from flies, ice fish, humans, and organoids, which I've never come across before, but mini guts, as I understand. So incredibly fascinating and interesting presentation. I'm sure you will agree. Professor Daniel mentioned that it was 10 years to find two pieces of the jigsaw puzzle, but I'm sure you'll agree that there were two absolutely vital parts of this jigsaw puzzle and help explain some of the most fundamental physiological processes. To Professor Daniel.
Thank you very much. Wow. Gorgeous. Thank you.